Hello, welcome to the RBR Works Cellular Respiration presentation for Honors Biology with Mrs. McDonough. Today's topic will be presented by Rishi Chudasama, Brody Ivy, and Rami Abdurrahman. Cellular respiration is a process in which cells convert sugars into energy. Cells require fuel and an electron acceptor to create ATP and other forms of energy to power cellular reactions. The first step is glycolysis, which glucose molecules are broken down. The second step is the Krebs cycle, where NADH, FADH2, and ATP are made. The third step is the electron transport chain, where NADH and FADH2 drop off their electrons and go through the transport protein. All these steps will be explained in more detail on the next few slides. The process of diffusion is where gases move from an area of high pressure to low pressure. Internal respiration is a movement in the internal tissues between cells and capillaries, and external respiration is when gases are exchanged between the alveoli and lung capillaries. Diffusion usually continues through the cell membrane until the concentration of a certain substance is the same on the outside and the inside of the cell membrane.
The first step of cellular respiration is glycolysis. The term glycolysis means sugar breaking, and it takes place in the cytoplasm. 2 ATP molecules become invested to start the process of glycolysis. Overall, 4 ATP molecules are produced for a ratio of 2 ATP per molecule of glucose. During glycolysis, 1 molecule of glucose, which is a 6-carbon compound, is transformed into 2 molecules of pyruvic acid, which is a 3-carbon compound. Those 2 molecules of pyruvic acid are sent to the Krebs cycle, but that's not all. 4 high-energy electrons are passed to the carrier, NAD+, to produce NADH. NADH then carries these electrons to the electron transport chain. As the bonds in glucose are broken and rearranged, energy is released. The second step of cellular respiration is the Krebs cycle. It's also known as a citric acid cycle. It was named after Hans Krebs, who was a British biochemist who proved and demonstrated its existence. It takes place in the matrix of the mitochondrion, and it begins when pyruvic acid produced by glycolysis passes through the 2 membranes of the mitochondrion and into the matrix. You can see that in the image to the left. Once inside the matrix, 1 carbon atom from pyruvic acid becomes part of a molecule of carbon dioxide, which is eventually released into the air. The other 2 carbon atoms from pyruvic acid rearrange and form acetic acid, which is joined to a compound called coenzyme A. Then, when citric acid is produced, it gets broken down into a 5-carbon compound, and then it becomes a 4-carbon compound after releasing 2 CO2, 2 carbon dioxide molecules along the way. Overall, the process of the Krebs cycle produces only 2 ATP molecules.
Alright, so step 3 is the electron transport chain. This produces the most ATP, around 32, which is much more than the 2 produced by glycolysis and 2 produced by the Krebs cycle. So the products from glycolysis and the Krebs cycle will go into the electron transport chain. Now, just to recap, glycolysis produces pyruvic acid, which is used in the Krebs cycle, and it also makes high-energy electrons. So when these high-energy electrons pair with a chemical called NAD+, they make NADH. So NADH is also similar to FADH2, and this is kind of like the main objective of the Krebs cycle. It's to create these sort of batteries. So now comes the main part. NADH and FADH2 will go to the electron transport chain to sort of drop off their electrons, and the high energy electrons will go through electron carriers in the inner membrane space. Now this is happening because when they go through the electron carriers, this is happening so much that there's a lot of other hydrogen ions already in the intermembrane space, and this makes the intermembrane space positively charged. So this will create a charge difference, and because of this charge difference, the electrons will want to get back to the other side to create an equilibrium. However, the only way they can get back to the other side is to go through a special protein called ATP synthase. So the charge difference will force hydrogen ions across the membrane through ATP synthase, and this force will actually rotate ATP synthase. This is a very interesting protein because it has this rotating mechanism, which will sort of grab an ADP molecule, which we know has only 2 phosphates, and it will attach another phosphate to the group to make ATP every time it rotates. So in conclusion, it will create 32 ATPs and expel water or H2O as a waste product. Alright, so anaerobic respiration is done when there isn't enough oxygen to continue the Krebs cycle and electron transport chain. Normally during aerobic respiration, oxygen would be the final electron acceptor, and it would combine with hydrogen to become H2O or water. But when there isn't oxygen, the Krebs cycle and electron transport chain can't function. And as we know, glycolysis is the only step that can function without direct oxygen input. So in simple terms, anaerobic respiration is when glycolysis is just done repeatedly. It's just done over and over again. And so glucose will go through glycolysis, which creates pyruvates. And in this process, NAD+ picks up electrons and becomes NADH. But at some point, NADH can't accept any more electrons. So without oxygen, there's no way to get rid of them. And this is when anaerobic respiration will kick in. So pyruvate will then become lactate or lactic acid. And this is where the electrons will go. So the electrons from NADH will go to lactic acid, resulting in NADH becoming NAD+ again. And now that we've freed some NAD+, it can go back to the beginning and start accepting electrons so that glycolysis can keep running. This is called lactic acid fermentation, and it's why your muscles feel sore after a workout. Your muscles run out of oxygen, and so they will resort to anaerobic respiration, and this creates lactic acid, and that makes your muscles burn. Now, something very similar happens with alcoholic fermentation, but this only happens in yeast. Basically, the pyruvate, instead of becoming lactic acid, it becomes ethyl alcohol. And in this process, it releases a carbon molecule, so CO2 is a byproduct of this process. And this allows glycolysis to keep running, same as lactic acid fermentation. And this is actually how a lot of beer and different types of alcohols are made. So, yeah.
In this chart, we see how cellular respiration is both anaerobic and aerobic. First, glucose is made into pyruvic acid when 2 ATPs are added during the process of glycolysis. If there is oxygen present, it goes through the Krebs cycle, which makes it also go through oxidative phosphorylation. If no oxygen is present, it turns into either lactic acid in animals or ethanol.